Well, that's a tough act to follow, and I know I've seen Robin's lecture many times, but I always enjoy it because it brings back those heady days, I suppose, when we were first starting to follow this exciting road along the Helicobacter connections. I called it my talk Helicobacter connections because the observation of bacteria in the stomach had been made before, although not as carefully studied as Robin's were. But by connecting up other bits of information in the literature and things we observed ourselves, we were able to develop quite a lot of confidence that we were correct early on. And so time and time again in the first couple of years, people were rather horrified that we were so confident that these bacteria were serious pathogens and that we were confident that antibiotics was the answer. And the reason was because we suddenly understood many different aspects of the literature and could read a lot more into it than people could see. Now I'll start by this quote by historian Daniel Boorstin, who says, "The greatest obstacle to knowledge is not ignorance, it's the illusion of knowledge." So this is where we start the story of Helicobacter connections. 1982, at a time when ulcers were common, the cause was known to be stress or genetics or smoking, and every doctor was an ulcer expert, and they didn't need to be told a new cause for peptic ulcer. And here let me remind you about ulcers. On the left, a duodenal ulcer. You see here, as Robin described, the first inch or so of the duodenum, and on the right, a gastric ulcer, usually on the lesser curve here. And you can imagine how these ulcers would become deeper and sometimes burn a hole into a blood vessel, and this has been a bleeding blood vessel here with some dark staining showing some blood. That's a duodenal ulcer. And so people could bleed to death quite easily from duodenal ulcers. And here's another one, a gastric ulcer, and sometimes these ulcers could become so deep they would penetrate through the gastric wall, and all the acid and food had come out into the peritoneal space. The patient would suddenly be struck down with a severe, excruciating pain, have a fever, low blood pressure, and probably die within 12 or 18 hours. So that was the typical thing that you could die from with peptic ulcer. It just seems impossible to imagine these days that people could tolerate walking around their whole life with any tick of the clock suddenly being struck down out of the blue with what could be a fatal illness. And that possibly tells us why it was possible to sell very expensive treatment to people with ulcers once you had a treatment. Now, on that vein, here's an article which was cut out by my father from Fortune magazine in 1983 and shows that SmithKline was pocketing $1 billion a year at that time in Tagamet sales. And Zantac was about to make the ulcer drug business even greater, ultimately selling $3 billion per year by the year 1990. But one statement in this article was wrong. It said that Tagamet cured ulcers, the green pill that cures ulcers. But that statement was wrong because these acid-lowering drugs healed ulcers, and the ulcers nearly always recurred once the drugs had been stopped. So the drug companies had invented a treatment but not a cure. But the great thing was the patients had to keep taking this drug forever. And so patients with dangerous ulcers were told to keep taking the drug in all kinds of different doses, but basically they could never stop taking the medication. So in 1981, we'd done some interesting activities. We'd studied patients with H. pylori, but we didn't really see any important new information from that. We'd taken biopsies from consecutive cases and from normal mucosa unaffected by a local lesion, so away from the ulcer. And this actually strengthened the type of information that Robin had collected. Then we started to search the literature, and we did find that there were many reports of spiral bacteria in animals and man. And so we could really follow this up and have some confidence that this wasn't just a very strange infection that was occurring in Perth, Western Australia, perhaps from wombats or kangaroos or something. So we could tell that this had been described in America over the past 100 years, for instance. And we tried to culture these things, Campylobacter-like organisms we were calling them, CLO. And we used what we said the Lee method, which is a microaerophilic culture necessary for Campylobacters. And Adrian Lee, who's here in the audience, had reported culturing spiral bacteria from the mucosa of the mouse colon. And so we were copying his methods. And in fact, we were using exactly the right technique from about a month after we started the work in 1981. But as Robin said, the months went by and we didn't culture the organism. Now, what about all these descriptions of bacteria over the past 100 years? Well, here's a classic one. And as you see, this is Helicobacter pylori, a beautiful electron micrograph image, sheathed flagella in the gastric mucosa. And this was from Susumu Ito, who was an anatomist, electron microscopist at Harvard University. And he biopsied his own stomach and discovered that he was colonized with a spiral organism. And this image was published in 1966 in a well-known textbook called the Handbook of Physiology. Now, Ito had studied these bacteria in animals and cats, where they're almost universally present without much pathology. So then he swallowed his own biopsy instrument and pulled out the biopsy and published it there. And he said, well, there you go, cats and dogs have them, and humans have them as well. But he didn't realise that at that time nearly every Japanese person in the world was infected with Helicobacter. It's almost universally present in Japan, the country with the world's highest rate of gastric cancer, by the way. So basically the problem after that was that people who studied medicine at Harvard University were probably told about spiral bacteria in the human stomach. And so you can see the conservatism that would then result if we said, we've discovered them, and they said, we knew about them. Professor Ito showed them the bacteria, showed us the bacteria when we were medical students. So what's new about that? So then we did a prospective study, 100 elective patients. And the reason we used to do 100 patients is because it's very easy to work out the statistics. 80%, 60%. That's true. And we had a prospective study. And the question was, how did these bacteria get there? Could it be that people were taking Tagamet, lowering their acid level and then being infected? Was it Kentucky Fried Chicken, increased global travel to Bali, different places like that, cats and dogs, drugs? All kinds of questions, as you can imagine, and Robin's correct. We had some pretty crazy questions on that questionnaire. Travel, pets, drugs. I didn't do sexual activity at that time. I was a bit shy coming from a Catholic school. Teeth, dentition. And so we said to the patients, how many teeth have you got? And how often do you clean your teeth? And we got some pretty strange answers, I can tell you. But of course, a person who has no teeth and never cleans his teeth has good dental hygiene. But his teeth have gone, of course. Endoscopy. We performed endoscopies and did the usual endoscopy report and classified things as ulcers, esophagitis, cancer, etc. We took biopsies of normal antral mucosa, and the histology was sent blind to Robin Warren, except if the patient had cancer or something. We had to tell Robin that it was a cancer case, but basically he was not told whether the stomach looked red or normal or bleeding or whatever. And the culture and gram stain section was sent blinded to the microbiologist who was then trying to culture the H. pylori. And he could see them on the gram stain, but again couldn't culture them at that time. So this is earlier, 82. And the results are that the bacteria were ultimately cultured from 12 patients, and the details are that the patient, the first culture was from a patient biopsied on Easter Thursday, 1982. The patient was number 37 was a 70-year-old male. He'd had a history of duodenal ulcer and gastric ulcer. He was anemic, he had an artificial heart valve, and he was on warfarin, anticoagulants. And so it was a major problem in this patient's management in his life to have duodenal ulcer disease. And he didn't actually, I think he did have a small duodenal ulcer at that time. And so the biopsy was cultured, but there was a methicillin-resistant MRSA, Staph aureus, epidemic at Royal Perth Hospital at the time. So the microbiology lab was very, very busy, and they were overworked. And so there was no time to check the culture on Saturday. It was not examined until Tuesday, and on that day gram-negative rods were seen in pure culture on this biopsy. And so they needn't have thrown it out after 2 days because in fact the stomach was sterile, almost, except for H. pylori, and quite a few of those non-specific blood agar cultures were just pure cultures of Helicobacter if you waited to the 4th and 5th day. Because as you can imagine, I was pretty annoyed about this as well. We'd been wasting our lives for 6 months. We'd been using the right methods and the cultures were being discarded after 48 hours, so we move on. The association of bacteria with endoscopic diagnoses. So obviously Robin told you the very strong association with gastritis. And then this struck my eye as I looked at the tables. All 13 patients with duodenal ulcer had the bacteria, 13 out of 13. So you can imagine you're tossing a coin. Half of all the patients had bacteria. And how often do you get 13 heads in a row? Well, the p-value on a single-tailed test, I think, two-tailed test, I think, is 0.00044. So it's very, very unlikely to be by chance. But you can imagine at a time when gastroenterologists are used to seeing hundreds of patients in clinical trials, they're trying to find the difference between an 85% cure rate and a 90% cure rate. To give them a revolutionary discovery on the basis of 13 patients from Perth, Western Australia, it was just not going to happen. Now, very interesting here, 13 out of 13 duodenal ulcers and 18 out of 22 gastric ulcers. And 4 patients had both types of ulcer and were all Helicobacter positive. But if you just had a gastric ulcer, it was 18 or 77%. So that was pretty interesting. But as Robin said, it was kind of suspicious because why do you get something down in the duodenum when it's a different type of mucosa? And yet we're studying the histology and the gastritis in the stomach. So it was a bit of an unusual finding at first. Now I remembered that I'd read something very interesting about gastritis, and it was from Magnus in 1952. And he looked at postmortems, motor vehicle accidents, anybody he could find. And he found quite a few that had some peptic ulcer disease of different sorts. And he noticed that in gastric ulcer, gastritis was present in 80%. But in duodenal ulcer, gastritis was present in 100%. Exactly the percentages we had for the link between bacteria and peptic ulcer. So it was, I don't know, maybe a fluke or something that I had, that had stuck in my mind. It was a paradox, and everybody had ignored Magnus's findings because they didn't fit in with what people thought would be the norm. And most gastroenterologists, I presented this in 1982 at a little, the College of Physicians meeting in Perth, and the people were standing up in the audience saying, Barry, you've got this wrong. People with duodenal ulcer don't have gastritis. And I said, don't they? Oh, I must have read that wrong. So, of course, I went back and checked my facts, and by the end of that year, I was dead certain that I was correct here. But the other interesting thing was gastric ulcers with normal histology were known to be commonly associated with non-steroidal anti-inflammatory drugs. There were a few papers in the Lancet in different places that could show that. And when you thought about it, maybe it made sense. In the stomach, anything you eat is directly applied to the mucosa. So you could have Helicobacter causing ulcers, but anything else you put inside your stomach could potentially cause ulcers, like non-steroidals. But it was quite hard for the non-steroidals to actually get down into the duodenum and cause an ulcer. So you might get a purer form of peptic ulcer down in the duodenum. The second thing is that maybe there was some reason why the Helicobacters would be down there as well, and that might be an interesting line of research. So in 1983, after much mucking around and talking to the editors and arguing over who was author, etc., we did publish our Lancet letters. And basically, we had a new species of bacteria, which is a bit like a Vibrio, like a cholera, and a bit like a Campylobacter, which is the bacteria on chickens. And the bacteria were linked to gastritis, and we knew that all the literature on gastritis involved peptic ulcer and stomach cancer. There was gastritis, and then there was 2 subsections, ulcers and cancer. They were both linked to gastritis. And since the new bacteria are associated with gastritis, as described by Warren, and this is the quote from my letter, then they may play a role in other poorly understood gastric diseases, i.e. peptic ulcer and gastric cancer. So we staked our claim at that point to this idea. But we're still a bit nervous and not really very confident about the whole thing. And we started thinking about it. So here you have all these people coming to endoscopy. That's the green group there. The green ones don't have Helicobacter, and about half of them are in the red group which have Helicobacter. But the interesting thing is nearly all the patients with duodenal ulcer, we studied other patients of course, and most of the patients with gastric ulcer were in this Helicobacter positive group. It was almost as if you could not get a duodenal ulcer if you didn't have Helicobacter. I mean, if you could, why weren't we seeing duodenal ulcer patients in the green group? So if you extrapolate back from that, what about if you could eradicate the Helicobacter? You'd move a patient from the red group into the green group where it's impossible to get duodenal ulcers. So therefore, on this basis, just from sheer logic, you should be able to conclude that antibiotics are going to cure ulcers. So this is where we were at the end of 1982. We're starting to get pretty excited about this, as you'd imagine. And I didn't have a job. So I went to the cocktail party after the presentation for the College of Physicians, and everybody else was drinking beer. I was drinking Coca-Cola, and I'm saying, where's the job, where's the job? And eventually I connected up with a fellow called Ian Hislop at Fremantle Hospital, and he said, this is interesting stuff, Barry, because I've been to the Mayo Clinic and studied gastritis, and I think you're wrong, but it's a curious finding and we need to look into it. So I then had a job in internal medicine and gastroenterology at Fremantle Hospital, which was not the main teaching hospital, but I did far more endoscopy down there than in the gastroenterology training scheme, it turns out. And Fremantle Hospital was a bit of a fringe hospital where you could do some experimentation and have an interesting life as well. Great place, Fremantle. Known facts about ulcer disease: the stomach was sterile. Ulcers were caused by lifestyle or stress, diet, alcohol, drugs, or genetically determined. So you see, the doctor who can't cure you is blaming you for having the ulcer. It takes the pressure off. Very comfortable. Our work was rejected because the results were outside the current paradigm, and the gastroenterologists had an investment in that paradigm, and I'll tell you more about that in a second. And so this was the pattern for the next 8 years. And over many drinks perhaps, Robin and I, and Wynne, Robin's wife, I think she would have been the first one to mention the Nobel Prize about this, perhaps in 1984. And she would say, well, when do you think you'll get the Nobel Prize for this? And Robin and I would say, oh, probably about 1986. So that was, Nobel Committee had the last laugh on that one. So, strange things are happening to my slide, but don't worry. Was H. pylori a pathogen? How do we test this hypothesis? Do patients have antibodies? And we soon found out that they did by doing some fluorescent antibody studies. So that was, it looked like the immune system thought H. pylori was a pathogen. Do antibacterial agents heal gastritis? So could you get rid of the bugs in some way? And we tested that and found yes. Have Koch's postulates been fulfilled? What is the disease process? How does it infect you? How does it survive in the acid stomach? So this is where we were going in 83, 84. There's those crazy little bullets. A new look at the literature then. Heavy metals were antibacterial, and arsenic and mercury had been used for spiral bacteria, syphilis, for about 100 years. Bismuth had been used to treat gastric diseases for 200 years. In fact, in Germany, it was invented many, many years ago. And it was one of the things that people would commonly take if they had stomach diseases. Now, the interesting thing, bismuth was also an ulcer treatment. It was called Denol. It was available in Australia, and it healed ulcers just as well as Tagamet, but did not lower stomach acid. So it worked totally differently. And it was thought that it coated the ulcer and protected it from acid. But the weird thing was that relapse after bismuth treatment was lower. So you healed the ulcer and you stopped the bismuth, and then a lot of the patients never ever got their ulcer back. And a few thoughtful gastroenterologists were saying, it obviously heals the ulcer much better than just lowering the acid level. But nobody really knew what that meant. And we said, well, hang on a minute. The only way we could explain this was bismuth killing the Campylobacter-like organism? So now we had a new hypothesis which we could quickly test. And so every week we were getting new hypotheses and sort of working on the weekend and doing all kinds of things. So here's the data that was in the literature then. And here's a study by Martin and Hollanders published in The Lancet 1980, I think. And here you see patients who are healed of their ulcer, and then we stopped the treatment, and they waited a long time to see if the ulcers came back. They had endoscopies every 3 months. And here you see the recurrence of the ulcer, and you see if the patients were healed just with cimetidine and you stopped the drug, they had a relapse rate of about 15% per month. And so that after a year, nearly all the patients had got their ulcer back. However, if the patients had been healed with bismuth, and it was a horrible medicine to take in those days, it was like windshield washer fluid or Windex. It had ammonia in it. But the patients who took it, they healed their ulcers, and then when they stopped the treatment, 60% got their ulcer back. But 40% didn't get their ulcer back. And the gastroenterologists that I showed this to, they say, oh yeah, but you know, if you wait another year, they'll get it back. But I said, well, hang on a minute. Mathematically, that's an exponential curve. And I reckon that's flat. And if you wait 100 years, it's still flat. So I said, I think that bismuth cures 40% of people, so it must be an antibiotic. So we did some studies at Fremantle Hospital, and this is probably the most exciting day of my life, when we'd put the bismuth disc on a plate of Helicobacter pylori on a Friday, and we came and opened the incubator on Monday, and bingo. There it was, a big zone. People had forgotten that bismuth was an antibiotic, almost, because it was just getting popular when penicillin was discovered. But before penicillin, bismuth was an important antibacterial thing. So when I saw that, I think that day, I thought we might win the Nobel Prize. And I probably called Robin up. And then I had an intern who was a terrific guy, and he agreed to take some bismuth. And here you see an hour after he drank the bismuth, he had H. pylori. And here you see the Helicobacters practically exploding with bismuth all around them. And so the drug that affected relapse rate was an antibiotic. So now we could use bismuth in our clinical trials, because here's the problem. If you did a placebo-controlled study of ulcer patients, you wanted to use cimetidine versus an antibiotic. But antibiotics were so experimental, you couldn't have people with a fatal disease, potentially, trying out something which might not work. They had to always have an active therapy. So that would be unethical. However, if we did H2 blocker versus bismuth, they were both ulcer healing agents, and then we could boost them with antibiotics or placebo in different ways. And we had H2 blocker antibiotic, bismuth antibiotic, H2 blocker alone, bismuth alone. And it was statistically a terrible study. You would never do it again. But anyway, we got lucky on that. So then, the story about the Koch's postulus and drinking the bacteria, and I'd say the title here is A Red Rag to a Bull, and I presented some of our histology data and healing of gastritis with bismuth to our pathologists. Now, Robin didn't mention it, but the p-value on his association with gastritis was 0.1006. It just came out as zeros when most computers did it. He had to use his Hewlett-Packard calculator to actually get this number and do the factorials. And I would present some of this kind of data and get the comment, Dr. Marshall, these changes seem very subtle. And I say, are you crazy? These commensal bacteria merely infect people with ulcers. So I said, hang on a minute, this data are the patients without ulcers. So, I mean, it was pointless. And I realised then it was kind of like a religion. Failed attempts to infect pigs. We did try to do it in animals and failed. I knew that from blood donors by then, 40% of the population in Fremantle had Helicobacter, and they seemed to be walking and talking and having a pretty good life. So I knew that it wasn't necessarily fatal to have Helicobacter. And I treated a patient with bismuth and metronidazole and had a cure. Great clinical response. Patients were trying it. Their GPs were giving me calls, sending me more patients. And we soon had quite a lot of experience that this was really a miracle cure for people with stomach problems. So the experiment has now been immortalised in this comic from the Abbott company who's selling the antibiotic for Helicobacter, of course. And so this is Neil Noakes at Fremantle, and he says, Dr. Marshall, you're crazy, and I'm saying there's no other way. In fact, it was a very lot of pressure on me because people were bleeding in my practice and dying from ulcers in my hospital. I could see it, and I was the junior registrar or senior registrar, but not actually in control of these patients who were under the surgeons and different physicians. And so I felt that there was an urgency to solve this dilemma and do the experiment. The only person in the world at that time who could make an informed consent about the risk of drinking Helicobacter was me. So I had to be in my own experiment, and if I submitted an ethics committee application and had it rejected, I probably would have done it anyway. And then I wouldn't be able to publish it, and I'd get the sack, and my life would be over, etc. So I decided to do it anyway, and I had some unofficial support from my senior colleagues because I had to have an endoscopy on several occasions. The chief of microbiology had to help me grow the bacteria, so there was emotional support or moral support, if you like. So we cultured a patient with gastritis. I underwent a baseline endoscopy. I treated the patient successfully. I drank the bacteria, 10 to the 9th colony-forming units. I had a vague illness with some halitosis. And my mother was the only person who told me I had a bad breath. And I said, you're just a silly old nurse, get out of here. But in fact, the problem with bad breath and halitosis, it is a medical disease, and some people are never told their whole life. And they'll be moved, if they're in sales, they're just ruined. So feel sorry for people with halitosis and tell them to get some antibiotics as quick as possible. Then I had this vomiting illness, no acid present in my vomit. And when I vomited early in the mornings, still half asleep, but it was just like water coming up. And as a medical student, I'd been to a lot of parties, and I knew that when you vomited, it was burning. So this was unusual. My father still hasn't forgiven me for my 21st birthday, but I claim it was the dog. So no acid present in the vomitus, and this was a key thing. And then I had endoscopy and biopsy and showed the Helicobacter. Histological gastritis was present, so Helicobacter was a pathogen. And here's Dr. Warren, immortalised in the same comic. He says, Marshall, your stomach lining is dangerously inflamed. And that's the first and last endoscopy Dr. Warren ever did in his life, as far as I can tell. And I'm thinking if only I had a simple quick test to diagnose the bacteria, then I would be spared this burdensome procedure. That was the day of my endoscopy, and I realised that if you had to have an endoscopy, this Helicobacter treatment would never catch on. We needed to have non-invasive tests, and ultimately we had breath tests and all that stuff now. So that was useful in my career to have it be on the receiving end a few times. And here's Dr. Warren, Gads, swarms of bacilli. You have a raging infection, Marshall. The spiral bacteria have taken hold. That was true enough. And so here's my biopsy. It was a bit fuzzy. But it was practically falling to bits. The mucosa was just like wet blotting paper. And these are those changes that Robin showed you. Very, very abnormal epithelial cells. No mucus, full of polymorphs, and no acid being produced in the stomach. And this is the silver stain. Very abnormal looking epithelial cells, masses of bacteria. So I was very excited. I came home and told my wife finally that I'd done the key experiment, and of course another very exciting day in my career. And she was pretty horrified at this time because she thought Helicobacter was a pathogen. She thought she'd be getting it any minute, and the kids, etc. So I was either having to move out of home or terminate the experiment. So on the 14th day we had another endoscopy, and I'd started taking antibiotics. So that was the end of it. But poor Dr. Morris in New Zealand, he had it for 3 years before he could get rid of it. Now, when I thought about this, gastritis, I'm catching an infection, no acid in the stomach. And it just at that time, my father-in-law, who can't be with us here today, he's 95, but he's still in good health, except having his knees done. He'd come back from London and he'd gone to one of the old bookshops in Oxford and brought me back Osler's Principles and Practice of Medicine, 1910. And I remembered reading it, and I went back to the chapter on gastritis and had a look at it. And Osler had these words: examination of the vomitus reveals, as a rule, absence of hydrochloric acid and sometimes the presence of volatile fatty acids. So Osler had described a very common paediatric syndrome which he saw all the time at the turn of the century. And in those days, when some children were vomiting, the mother would save the vomitus in a bowl. And the GP would come around. He'd put the litmus paper in the vomitus. He'd say, absence of hydrochloric acid. This is achlorhydric gastritis. Don't worry, it'll get better in a few days. Your kids will be okay again. They'll get their appetites back. But we now know that was the precursor of Helicobacter, and they had the organism then for the rest of their life, and sometime in the future they're going to get peptic ulcer, and maybe when they're 70 years old they get stomach cancer. So that's the acute attack of H. pylori. It lasts a few days, resulting in lifelong colonisation of H. pylori. Then how does H. pylori survive in the stomach? And Robin and I used to discuss this often. Now, it's because of urease enzyme. Gastric urease was discovered in 1926. It was one of the first enzymes ever isolated in crystalline form and it was used to prove that enzymes were proteins. The association with H. pylori was noted by other people in 1984, and H. pylori made urease. And I did some work then on patients and showed that urea was absent from the gastric juice of patients with H. pylori. And I knew that H. pylori could split urea to make ammonia and bicarbonate, so obviously maybe this was H. pylori protecting itself from acid. And we did an experiment. Basically, you could take the car battery acid out of your car battery, dilute it 10 to 1 with water, add a speck of urea, and tip H. pylori in and stir it up, and H. pylori would swim happily in that car battery acid for 30 minutes, and every other bacterium in the world would be killed, but not H. pylori. You can still culture it out of acid. Then we did the biopsy urease test, which was as accurate as a gram stain, took a few minutes, and anybody now could diagnose H. pylori. So it got very exciting for gastroenterologists again. They could do a biopsy and they'd have the answer to the patient's ulcer problem in about 20 minutes usually. So implementation, we then had effective therapy, bismuth and antibiotics, and ultimately proton pump inhibitors, Losec, with 2 antibiotics turned out to be the best possible treatment. Once you got rid of the acid level, then the antibiotics such as amoxicillin would work in the stomach. I and others worked on non-invasive breath tests for H. pylori. So nowadays, GPs can treat H. pylori. They do a blood test, you've got H. pylori. Now I'm going to give you antibiotics. Now I'm going to do a breath test to see if the antibiotics worked. The poor old gastroenterologists are out of the loop, you might notice. So Robin and I claim that we're actually in the business of curing colon cancer right now because those gastroenterologists need something useful to do. And they're out there doing those colonoscopies instead of looking after ulcers. Non-invasive follow-up is possible. Bye-bye gastroenterologists for ulcers, anyway. So validation followed that. We did a double-blind study in 100 patients, treated them with different combinations. We got a cure rate of 80% versus 10% in the control group. And this was repeated by others. Rouse and Tightgat in 1990, David Graham in Houston, 1991, and Henschel in Vienna in 1993. And Henschel's was the cleanest study and was published in the New England Journal, and that's his data. Here's the relapse in the patients given the Tagamet plus a placebo, and this is the relapse in the patients given antibiotics plus Tagamet or something like that. But basically the antibiotic patients were cured. So the whole thing was accepted really in 1994 at the National Institutes of Health Consensus Conference, and you'll be pleased to know my talk is now finished. Postscript, Nobel Prize in Physiology and Medicine. Thank you very much.